Okay, let's pull up information here. Hello, everyone. So we'll start here. I know it's not the ideal location to do a review overall, but I'll do my best. If you have questions there, that's fine. But something I'll do is I'm going to go through the calculations list as well, okay? So I'm going to pull up some of these. Oh, that's the rubric for a PowerPoint presentation you guys aren't part of. Okay. All right, let's pull that up. Final calculation problems. Final exam study guide. Okay. So these came up yesterday. Let me zoom in here so you guys can see. Okay. All right. So this is the final exam study guide. Remember that the final is comprehensive, so there are two other study guides that should be used to answer, to focus on questions that we've been asking. Okay, so make sure you do go to those and understand how to use each one. But this is just what we've covered recently, and what some of the stuff here has been covered in the most recent homework. So just paying attention to those pieces, okay? All right. Do we have people here? I didn't send out a reminder for today. It's not a required one today. I am recording it. It's going to be posted later. I'll go through the pieces that I can. One of the things is that for a few of these parts, because I know that just looking at a screen is not as useful, I'll turn things to the side and I'll work through a couple of practice problems if you guys need me to. Okay, Mavis and Angie are here. Good to see you. Good to see your usernames. All right. But again, this is a review session, and this is our last session on Twitch. And again, in this setup, it's a little bit easier for me to post it. I like the setup here, but I don't get to interact with you as audio oriented, okay? All right. So let's walk through a few pieces of this. The last last segment here, I think has not been quite as confusing. But I will, okay, yes, I will repeat that again. The final is going to be opened on Wednesday, and it will close on Thursday. So Thursday is when we are scheduled to take the exam. I can't have it opened on Friday for you because I'm required to turn in grades by Friday. Okay? So just understand that concept. All right? Thursday is when we would have normally had the exam. So I'm opening it up on Wednesday, and it's going to be open all day on Thursday as well. You may take it at any time, but obviously you'll have to work around your other exams. I'm hoping that no one's going to actually have a live exam. I think that would be very frustrating to try to work around people's schedules to do the live exam. I know you already had this on your schedule, but people's schedules are very different in the pandemic. I know that for sure. Okay. So again, you have 48 full hours. You have 2 hours and 15 minutes to take the test. And again, it's mostly going to be short answer, fill in the blanks, and a lot of multiple choice, but I'm going to have rotating multiple choice. Okay. And gonna try to do my best to make sure that you guys have unique tests, but that can be difficult, especially with the calculation ones. So we'll chat about what kind of pieces I'm going to put in there. Obviously, you're not going to have every single, you're not going to have such a focus on a certain type of calculation, but all the calculations that we've gone through will be in there in some way, at least part of it. Okay. All right, let's start with the gene expression, and then I'm going to move the calculation ones after we've gone through a few pieces of these. Again, use this as a study guide. My suggestion would be, honestly, write down for yourself each of the definitions for all of these. So you can have this study guide up next to the exam as you're taking it. I don't mind. as long as you're able to, let's say I ask you, what does CAMP do? You're going to have to be able to explain that to me, okay? So my suggestion would be to define and give functions for each of the pieces here. All right, so gene expression in bacteria. Again, you've got structural genes and regulatory genes. Remember, the structural genes create a protein. These are all still genes, but the structural genes create proteins that are used to build or have enzymes or some sort of thing. The regulatory genes regulate, actually attach to parts and regulate expression of genes, okay? What does it mean to have something be constitutive? Constitutive, remember, that is when it's in its default position. What is its default position? And that has to do with the inducible and repressible genes, okay? Inducible, a positive inducible means that it's always on. A positive repressible means it's always on. A positive inducible means it's always off, okay? A negative repressible means it's always off. A negative inducible, a negative repressible means it's always on and can be induced to be off. Hold on, I might be confusing myself here. We'll pull that back up. But the concept here is what do the various things do, okay? If it's negative, then the protein that is produced is a repressor. If it is positive, then the protein produced is an activator. And so a positive repressible is one where the activator is always on unless it's taken off. A positive inducible is one where it's on only if the activator is turned on and added, okay? The negative control, negative inducible, so let's say negative repressible. Negative repressible means it's always on unless the repressor is created and turns it off. A negative inducible means that it is always off unless something takes the repressor off. Negative inducible is the lac operon. Okay. And then there's that whole catabolite activator protein. And in large part, that one is due to helping bacteria use glucose preferentially. If lactose is present, it's going to turn on the lac operon, take the repressor off of it. Okay, remember negative inducible means that you have to remove a already stationed repressor. But the negative, it's going to decrease transcription if glucose is available, because it'll turn on, take that repressor off if there's lactose. But if there's lactose and glucose, it's going to turn off the lactose until there's not enough glucose, the lactase. Okay, so those kind of concepts is mostly what we covered here. Okay, so I'll go through more clearly. Hopefully everyone understood that. The main things here is that in positive control, the regulatory protein is an activator. In a positive repressible, the activator is always on unless something takes it off. In a positive inducible, the activator is always off unless something induces it to turn on. Okay. So you're adding the activator in a positive inducible. You're removing the repressor in a negative inducible, and it's the opposite for the others. Okay. So run through also and make sure you know all the components of the lac operon. And one of the key things here also is just understanding what happens if various parts of the operon are broken. If the repressor gene is broken, then it's going to be on constitutively. So what do mutations do basically? Okay, so understanding those pieces. All right. The gene expression in eukaryotes. So this was for the gene expression in bacteria. Gene expression in eukaryotes, the major pieces I want you to understand is that eukaryotes have those extra regulation layers, chromosome remodeling being a major way that we control what's transcribed, and that promoters and any promoter influences will strongly influence transcription as well. And then one of the key things we talked about was that RNA surveillance. The idea of when RNA surveillance and that was the nonsense-mediated decay. So if you see that there's an error that makes a nonsense mutation far up, basically you would know that RNA surveillance would happen. So if I told you that that's what happened, then you would indicate how that would be regulated by the cell. Okay. And then a lot of focus on the molecular genetic analysis and biotech. We talked about restriction enzymes. We talked about the various ways that genes are cloned, the cloning vectors, primers and how they're used, PCR, how reverse transcription PCR works, reverse transcriptase and how it's used in that. So there's a lot of pieces here, okay, that we've gone through in lecture. Again, go back through and mark out where exactly all of those are occurring, okay? These single nucleotide polymorphisms that happen outside of a gene, they aren't mutations because they don't impact actual transcribed genes. But what they are is they can be linked to a specific pattern of disease inheritance. So it's kind of a way of testing for the genes without actually transcribing, without actually reading the entirety of the genes. Okay, so all these have to do with DNA tech. Okay. One of the key things here that I want you to understand from this one is understanding when something is cut. Will it make a sticky or a blunt? So looking at those patterns, remember that for it to be sticky, it has to have these complementary sequences that are backwards to each other. Okay, so it's going to cut and have a sticking out end and a sticky end so it can stick together. And again, those the palindromic sequences for the sticky ones in particular. The gel electrophoresis, one of the key things that has to do with why gel electrophoresis is used for DNA technology is the fact that it can separate the smaller and larger ones, and we talked about that with the repeated patterns and how you can tell how long the repeated pattern is and how many copies of each one you have, and that's really how the DNA fingerprinting works. I encourage you guys, I gave you a very easy class participation. You go through basically a DNA strand. You find all of the repetitive sequences, and you cut at those sequences, and then you cut out those chunks, and then you look at those patterns, okay? Also, talking about the cloning and the insertion of genes of interest into plasmids, those experimental plasmids and what they have in them. Remember the blue-white screening to make sure the plasmids were made properly. That disrupts that LAC-Z, makes it so that it can't process a blue dye. And then you have the one that checks if the bacteria itself took up the plasmid. Check if the bacteria did. You have that extra selecting gene on there, like an antibiotic resistance gene, and only the ones with the plasmid will be able to resist antibodies. And then, of course, you have the gene of interest that you want to insert into, either use the bacteria to be factories for, and that's something like creating insulin or the Humira drug that I'm on, using bacteria to be protein factories, or actually inserting it into the a germ cell line of another plant or an animal, and inserting that plasmid DNA into them. Okay. There's a lot here. This has been the last week and a half. So hopefully you guys got most of this. It's all in the chapter 17. I posted that. So I have one folder for that. There's 2 lectures and an activity. Again, I recommend you go through the activity. It kind of shows you how the DNA fingerprinting works. It uses VNTRs as the DNA fingerprint. Again, STRs are often used, but either one can be used. Okay. So one of the key things that's part of the calculations is for this is that if you have a plasmid that you know the length of, you'll be able to tell if you have complete digestion, how long each one of the you'll either be able to tell me exactly the length, or seeing the length, you'll be able to tell me which restriction enzymes were used, if you know where the restriction enzyme sites are on the plasmid. Okay, so either you'll be able to use the gel electrophoresis to indicate when 1 was used, when 2 were used, or when both were used, or when 1 was used, when the other was used, or when both. Okay, so they have different patterns there. And tell me how long each of the fragments are. Those fragments are measured in base pair lengths. Okay, since plasmids are reasonably small. Also, we talked about using DNA profiling to determine if the sample belongs to a particular individual. That has to do with the video that we watched in class. And we also talked about parent-child relationships. In particular, you inherit some of these variable lengths from your parents. So one parent will have so remember, you have 2 chromosomes that have those short repeats, the short tandem repeats. So your mom has 6, your dad has 12, and you're going to have both of those lengths on the actual blot. And if your mom shares one with you and your dad shares one with you, then you can identify them as parents. Okay. If you go to the activity that I shared, there is also there's a participatory piece. You can do you can do parental testing through that, and paternity testing. And so with the paternity testing, you'll be able to actually look at the 2 pieces and say, okay, who's the father? Who could be the children, kind of thing. Okay, so I'll give you some practice. And then finally, that Sanger dideoxy sequencing, remembering that the way that this dideoxy sequencing works is that if you have these DDNTPs, they're making a fragment that adds 1 nucleotide and then cuts it off, and you see when they were added. So you see that this 1 that's marked was added at the 1, so the A that was marked was added at the 1, and so you just read up into the longer and longer. Okay, so you read from the bottom to the top. Okay, so there's a lot here. I'm realizing you might not actually be able to hear me very well because I took my microphone off, so I apologize if it's been quiet. Might actually be better now, so my apologies here. I didn't realize I still had it connected. Hopefully I've been talking loud enough. But this is on, so I realized that I should have had this on the whole time. Oh well. Okay, but you guys all said hello to me, so at least you could hear me a little bit, so hopefully it hasn't been too quiet. It was sitting next to me, so at least it was picking up some of it. Okay, that is it for the final study guide, but the big thing I wanted to go through today was this calculation list. I know I just posted it, which is why I want to go through it, okay? All right. So this gives you an idea, again, I'm going to zoom in just a little bit. This gives you an idea of, for the different chapters, what kind of puzzles or calculations do you have to work through, okay? So, there's the and or rules, and there's, so the independent assortment. You should be able to use the and rules to calculate out large numbers of pairings. So if I gave you, A, B, C, D, E, F, and I asked, and they're all heterozygotes, it's 2 heterozygotes for all 5 of those, what's the chance that you're going to get a big A, big B, little b, etc. Okay, all throughout. So that's what you need to be able to calculate out for that. Also chi-squared, in particular, looking at phenotypes, so being able to tell, how to tell the expected. The expected is going to be, say, for a dihybrid cross of 9 to 3 to 3 to 1. For a X-linked, you're expecting to see, 1 male, it's a 3 to 1 ratio, and most of the males, etc. Okay. Again, these, we talked about the mono, di, and trihybrids and frequencies and phenotypes. Interpreting pedigrees. I know some people had some issues with that before. So let's start here. Since you guys are all here, what I can do, I have my, there are going to be a few more questions on those at the end. Yes, I'm going to focus for the last few gene regulation and the, for that, I'm going to have a few more questions than on the other ones you've been tested on, but again, it is comprehensive. So let's see, we have had about 17 chapters-ish. I know some of them we didn't focus on as heavily. So once we focused on heavily, like 9 and 10, I'm going to have questions from both of those in the same frequency, because we went over those reasonably fast. Chapter 8, we had mostly focused on to understand how bacterial transmission works. But for the ones that we focused on, I'm going to try to make it reasonably equal between the chapters. For chapter 15 and chapter 17, there will be a few more. I can't work out percentages in my heads right now, but yes, there will be a few more. Okay, but not like a large number more. We're talking maybe for each one of those, for each one of the other ones. Oh, let's see. So maybe there's going to be like, let's see, 5% of 50. I'm not going to work out all the math right now, but that's the basic concept, okay? You may have like 10% instead of 5%. The final is cumulative. So it's going to be all the possible ones. This is why in the calculation list here, I go back to chapter 3. You will still have to know how to do chi-squared. And for some of these chapters, I'm going to give you calculation problems for each of these sets of calculation problems. You're going to have a calculation problem that comes from chapter 3. It could look like a variety of things. The idea is that if I gave you a chi-squared question, I could ask you about a chi-squared question for a more complex problem that has to do with, say, lethal genes or multiple alleles or epistasis, okay? So there's some complex pieces you might have to work through with chi-squared. So some of these could be combos, okay? But you're definitely going to have things that have to do with and or and the chi-squared for sure. So these frequencies that you'd have to work out using the and or rules, it's going to vary, okay? So I'm not going to tell you which ones you're going to come out with, but know how to do those, know how to build the problems and solve them, okay? Similarly, know what to expect from epistasis, what to expect from lethal genes, be able to explain complementarianism, complementation analysis, and kind of how complementation can happen in humans. That really talked about that blonde hair issue. The patterns of inheritance for sex-limited and sex-influenced. Okay, so if I tell you a sex-limited trait, remember that sex-limited, no matter what the genotype is, only one sex can get it. Okay, a male could have the genotype to create to have hyperovulation, but because there's no ovaries, it won't happen. Okay, so that's sex limited. Sex influenced means it works like a dominant in males or a dominant in females, and it works like a recessive in the other sex. That's influenced, not limited. Females can have pattern baldness, but they have to have 2 copies. All right. For these, though, okay, so let me run through what they are. And something I'd like to do as part of this is walk through how to do some of the problems, okay? I know that there is pieces that everyone has struggled with, so my suggestion would be pull out a pen and paper as I walk through what the various pieces are. You will need to be able to do those problems. So let's say, in terms of this one, say for chapter 4, I could give you an incompletely dominant trait and a codominant trait with multiple alleles. Maybe I ask you for an incompletely dominant trait that I ask you about their inheritance with a blood type, or I ask you about a simple dominance plus a lethal recessive. So being able to combine any of those types, and it doesn't have to be epistasis. It could just be a codominant with multiple alleles plus a gene that's lethal, and then having to understand what your outputs will be. Okay, so that's the same thing here again, being able to predict for epistasis, what kind of a output will you have. Again, from chapter 4, you may have, there's going to be like 5-ish questions from chapter 4, but it could be any combo of those inheritance types. I could put a codominant one with a X-linked trait. I could put multiple alleles with lethal genes, etc. Okay, so chapter 4 we will focus on a lot of those combos, the interesting patterns that can occur if you have genes that affect each other, like an epistasis, or genes that are paired and influence you don't get the 9 to 3 to 3 to 1 patterns because the patterns are different for 2 of them. Okay. All right, chapter 5. The sex determination patterns. Again, we talked a lot about the traits that people would have, and what is trans, and what is how this influences phenotype. But the actual calculation, coordination problems are more about determination, okay? In particular, that fruit fly problem, the ratio problem, okay? So expect to see something like that ratio problem for the chapter 5 questions. These are just the calculation-based ones that you might see where you have to do fill in the blank. Okay, so there's other questions that are going to be from these chapters. Note, I'm not only putting calculation on there. But again, with chapter 5, you may see a couple of questions about, about exactly how environmental sex determination works, or why Freemasons exist, etc. Those aren't really calculation problems. But something about this is one that you might see that comes from chapter 5. An XXY male and XXX female, and then maybe they both carry one trait, and the likelihood they're going to pass that down, and some complication in that. Okay, so that's an interesting one. In particular, the XXY male being able to pass on either one of his good exes, or an XXY male being able to be a carrier on one of those, or the complication of those mosaic calico males, XXY calico males. So those kind of concepts there. Again, those are the combo pieces I'm talking about that you may have to be able to put together multiple concepts. Chapter 6, again, this is one of those ones that you can solve for the aneuploidy. Again, we mostly talked about what causes aneuploidy, the results of a lot of the aneuploidies. Those are not calculation pieces. Those are studying the process of aneuploidy and how that happens. But you should know the meiotic stages. And in particular, the one from chapter 6 you may have to cover here is, in terms of a calculation, is in this combo of people, someone with an aneuploidy and someone without, or in this combo of who gave the error, you can solve something like, if there is a, say, XXY person, then you know that, then you could figure out based on pieces who gave the aneuploidy, because you'd be able to see, person. So if it was an XXX, you know that the woman must have given that aneuploidy, that because she's the only one who could have given two X's. If it's an XXY, then the male gave the aneuploidy. If you know that they have, say, if it's an XXY male, then potentially it could be either. So figuring those things out. All right, the next part that comes up with a large number of length of calculations is that linked gene, okay? Understanding how to do the notation for them, cis versus trans orientation on the heterozygotes, what you would expect for offspring from someone that has a cis orientation versus a trans orientation, and why that's different, okay? What the most common non-recombinant ones will look like. And then in terms of gene mapping in particular, being able to identify what the distance from the genes mean, do the 2 gene mapping, okay, if they're 15 map units apart, what patterns would you see in these phenotypes? And then the difficult one that I know we've worked on a bunch, the trihybrid crosses. Find the middle gene. Find the distance between each gene. Identify which one's the double crossovers. Talk about the coefficient of coincidence and then the interference. Okay, remember, coefficient of coincidence is how many expected crossovers, double crossovers happened, and the interference is what percentage or what frequency was prevented from crossing over because 2 crossovers interfered with each other. We've done a couple of the somatic cell hybridizations ones where you have to identify the chromosome of interest based on information about where is this, where is it not, and the deletion mapping as well. Okay, so any of those are more less calculations, more how do we solve this specific puzzle. Okay. Chapters 8 through 13, there will be questions on the final about those. There's no calculation problems. It's mostly memorizing processes It's mostly understanding how gene expression works and inside the cell, all the processes to actually turn genes into proteins is what 8 through 13 talk about a lot. 8 talks a little bit about bacterial. It talks about bacterial genetics a bit, and all we covered there was kind of the setup for understanding horizontal and vertical transfer and such, and how horizontal transfer can be used for chapter 17, biotech. Chapter 14, again, not much there, understanding gene mutations and what it does to proteins, but that's the only sort of calculation piece there. Chapter 15, understanding how the operon functions and where each piece clicks in, all the predicting where the mutation would have occurred if something doesn't happen that you expected to happen, sort of putting the pieces together, identifying where things must be mutated, and then understanding when catabolite repression would occur on the lac operon. Chapter 16 doesn't really have any calculations. We just ran through more of that gene expression that we talked about in chapters 12 and 13. And then finally, the only thing for chapter 17 that was really, and we already went through that in the study guide, understanding how to do the restriction mapping. If you know the length of DNA that you are cutting and you know where the restriction sites are, and you have multiple restriction enzymes you're using, be able to predict the length of the base pair lengths of your DNA on the gel electrophoresis, okay? And then how to read the Sanger sequencing, how to do DNA fingerprinting. Okay. I went through all that reasonably fast, I know. What I want you guys to tell me is where in this calculation set of since the beginning have you felt like you need more guidance? What I can do is, after you tell me that, I will make something up and we can run through how to put together and solve that problem, okay? What I can do here is, obviously I need to snooze this. Okay. What I can do here is actually make me bigger. Okay, so once you have a question, I'll make me bigger, and you guys can, and then we'll work through it on that. Okay. Okay. Who would like me to cover any of these pieces? I know it's a lot, but that's what I'm here for today. We are reviewing the puzzles we've been putting together for this whole time, understanding those different pieces. You know, think about any of the questions you got wrong so if you got a low score on the first one, perhaps there's some of the problems that you just felt lost in. You didn't really understand how to do the X-linked problems. You struggled with the mapping on the last one. Okay, so let's walk through, for exam 2 in particular, how to do the trihybrid crosses. Okay, and I did put it in the last thing, so I'm gonna let me actually go here and pull that up. 1 second. Okay, give me a minute to open a few things, and I will walk through it, okay? But we can do even one of the versions together that was on the test. Speaking of which, can you guys see the test? Can you see your answers on the test? Can you see the Now, I know that for the fill in the blanks, I didn't explain it. Okay. Let me fix that right now, actually. I'm going to go there real quick to here so you guys can see your actual results. So what's going to happen, because this is open book, and because I may be using some similar questions, you won't be able to access or see your exam 2 during the period of the final. Okay, I'm going to make it unavailable as it is today. But until then, I will make it available so you can see correct and incorrect answers. So let me just go here real quick to course content. Go to your online exams. I'm not showing you that I'm doing it, but edit the test options. What I'll do is I'll make it available. Okay. After the due date, and I'm going to change its availability, okay? So the availability, I'm going to change until, so it's available to you until Tuesday. Next Tuesday, not this Tuesday. All right. So I fixed that now, okay? What you're going to be able to see now is the actual test itself. You're going to be able to see the pieces to put together the test, okay? So the concept here is you'll be able to see what the possible answers were and what you got, what you answered and what the actual right answer was. Again, this will disappear before the final is out. Okay. All right. Let's walk through. I'm not going to pull this that up on screen, but I will pull up one of the options for exam 2. All right. I could probably I'm just going to begin the test on my end. It's not like it actually grades it. Okay, we're going to go over here to the actual 3-part. Okay. Let me put this here. And I'm going to turn a few things around, so hopefully no one gets dizzy. Here we go. Okay. I was working with other stuff with the students today. Okay. My 10-10 class finally went through this stuff. You guys know this. All right. It's been all genetics all day today. Okay. All right. Let's pull this up. So, I know I've blocked most of the screen here. Sorry. So, this is the question, okay? I'm going to put it up on the board here so we can see it. Okay? Again, these numbers varied for everybody. So, we have light straw, type, light, and straw. Okay. Oh, no, wait. That's not the one. That's just a single. That's just a double. I have the other numbers here. Is that it? Hold on. No, where's my actual numbers here? Oh, okay, here it is. Sorry, my fault. Okay. So once more, I'll just make it. I started with the wrong one. Okay. So, hopefully I'm big enough so you guys can see still. Okay. Let's take off the light straw one. That was not the one. Okay. one of the key things we need to start with as part of this, we need to start with, what do the parents look like? Okay, remember, we always have these are the expressed, this is the phenotypes, okay? Remember that since you cross with a fully homozygous recessive, okay, and she needs chi-squared. Let's work through this one first. Fully homozygous recessive for all of those, that whatever dominants are there came from the parent. Okay, for this one, again, it does vary. I switched these around for a couple, so you got to make sure that it's not exactly what you saw. Okay, SHCBZ. Okay, these are the dominant traits. Okay, there's two of them that look alike. Okay, one parent. Okay, so that is the these are all dominants. Okay, and so the parent looked like this. Okay, dominance over recessive. That's what the parent with dominance looked like, because half of them, so we have the okay, half of them look like this, half of them look like this. Okay, so that is your dominant. Now, what do the double crossovers look like? This isn't in the right order. Okay, so what you first start out with is, what's the middle gene? It has to be SH, because you look for the one that's different than the parental types. Parental types look like this or this. What has shifted in the least similar ones? It means that the side ones have come up. It's actually in this organization. SH is in the middle, because SH is the one that has shifted in the two, very small number, in the two double crossovers. Okay? You start with that. SH is in the middle. I'm just going to type it out. Okay? Which genotypes of the double crossover? Once you know that, it should be the, again, if you put it in the right order, I gave you the credit, but the idea is that it is the number 3 and the number 7. You know those as double crossovers, they're the least likely. Okay. How do we find the distance from SH to C? Okay, the next thing we do, how many total do we have? We have 1,000, okay? In this scenario where it's set up like this, okay, a crossover in this space would move the C. A crossover in this space would move the BZ. Okay? Now that you know that, you can reorganize all of these, but I'm going to point them out. Okay? The way you find the distance from SH to C is, first, take the, we look at the ones that are single crossovers, and then we add the double crossovers to them, and that's the frequency at which these are both happening, okay? Every crossover in that region, okay? So, we take, I said that SH to C, this is the distance I want currently, okay? To do SH to C, you look for the ones where the C has moved, okay? That's going to be 6 and 1. Wait, hold on. Not 1. It's going to be 6 and 5. There we go. See? 2 dominants, 1 recessive. 1 dominant, 2 recessives, okay? And that the C has moved. This is that distance. 13 plus 15 plus the double crossovers plus 2. So that is 13 plus 15 plus 2. That's 30. 30 over 1,000 equals 1, 2, 3. 3 Mach units. Okay? Do you guys see how I did that? You had to find the middle one. Once you had the middle one, you organize it and say, okay, when there's a crossover between the C and SH, C moves. I look for the one where C is different than the parental. And then once you have done that, you add those two together plus the double crossovers. 3. Next. When SH and BZ, when BZ has moved, now we are looking at the distance from BZ to C. We'll get there. Let's move to, instead, BZ to SH, because that's going to be the one that's easier to do. To get the distance, the full distance, add these two numbers I'm going to create up here, okay? Let's start with this one, okay? So we have 3. SH, BZ to SH, okay? Remember, SH is in the middle. So the one where BZ has moved and nothing else has, that's the one, okay? BZ has moved in 7 and 1. All right, those are both 9, that's 18, 20. Next, add those together to get your full distance from BZ to C. Okay, next, what I need you to do is expected frequency of double crossovers. Multiply those two together. 0.03 times 0.02. Terrible doing decimals in my head, but you should have a calculator, okay? Expected. Now, what is the actual? That's how we're going to find interference, okay? The actual is 2 divided by 1,000. .0002. Okay, to find the difference between these, what percentage actually happened? About 33% of the actual expected ones happened. Do you see this? So about 1/3 of them. 2 divided by 6, you show that 1/3 actually happened. It was 2/3 interference, which you should represent it as a number, so you could have been 0.6666%, or because it's 0.66666, 0.67 or 67. Okay. Did you follow what I did? That's the process, okay? This is how many happened. This is how many should have happened. What percentage were blocked? About 66% of them were blocked. 67%. Get close and I'll give it to you, okay? All right. Faye, how are we doing? Did you understand what I did? I think the key is if you didn't figure out SH, you can't create your map. If you don't understand, if you don't have this, you can't find the correct ones. You have to know which ones moved. Okay, where are you stuck? Because there's a lot of pieces. Do you know how to find the middle gene? Let's start there. Where are you stuck? Can you find the middle gene? You're asking me to go over frequency and what kind, Kim? Okay, the double frequency. Okay. All right. Last part here. How do we do this? You start out by saying, if it was as simple as 0.03 times 0.02, if that was the simplicity of it, if double hybrids happened, if double crossovers happened and the arms didn't interfere with each other at all, then we would have seen these, we should have seen 6 individuals. Okay? 6 out of 1,000. We saw 2 out of 1,000. That is 2/3 less than we expected. That's the interference value. Okay, that's the frequency that, so again, this is the AND rule. The chance of it happening here and the chance of it happening here, that is the frequency that it will happen, that the doubles will happen, okay? You have 3 mu, 2 mu, which equals 0.03 times 0.02, 0.006, 0.002, 0002 is what you found. Therefore, it was 2/3 less than you expected. Therefore, the interference, chromosome arms interfered with each other 66% of the time. Okay? You're looking at the difference between just these two numbers multiplied together, then the others, okay? All right. Faye, we can do a separate meeting together. Send me an email for when we can schedule. I don't have a ton of time, but we can find a time to talk for about a half hour, okay? All right. Let's move to chi-squared. Where's my wipe? I'll just use this old towel instead. And remember, I am recording it, so you should have seen. You'll be able to go back and pause me, okay? All right. Next, we need chi-squared. Okay. Let's do a chi-squared for a more complex one. Let's say I give you one where you have a double hybrid, okay? So, I don't know, AA, BB, CC, okay? Sorry, that's not 3, let's do double. It's too many for the other, okay, so. There are 4 possible phenotypes from this. Okay, this is I'll move in a second, sorry. Okay, these are the 4 options. Displays both dominants, displays one dominant, displays the other dominant displays full recessiveness. Okay? For this one, what's going to happen is that if I told you there was double hybrid, and I told you that you needed to find out if the numbers I give you are indeed what you expect. Okay? So I would give you something like, I don't know, 100 individuals. I'm making up numbers, okay? Don't quote me on these things, okay? But we make up numbers and we say it's like 90, and then 55, 43, and 12. Okay? Question is that actually what we expected to find? So what you would do is you'd add these numbers up. Okay? 90 plus 55, 145, 180, 188. Somehow I made it end up be 200. Let's make it 200, okay? There's 200 individuals. So in this crossing, this is what you found. What did you expect? What frequencies did you expect, I guess? What frequencies did you expect? Who can tell me? Let's forget about the numbers. Just tell me blank out of blank or a percentage or a decimal. What should this be? Not 25 for each one. Okay, so that's the key here. You have to build it first. In this crossing, if you made a big 16 square, chi-squared, how many of them would have this expression? Both dominants. In that 16 square, how many? This is the most important piece for chi-squareds. It's not always 25% each. Sometimes it is. But when I give you a problem here, I'm asking you the probability that I would find two dominants that come out. If you did the actual pairing here, you would find 9 out of 16 times you have both dominants come out. All right? Because it's the combo of big A, big B, little b, big A, little a, big B, little b, big A, big B, big B, several different phenotypes, and you add those all together to create the 9. Okay. 9 out of 16. Okay. What is 9 out of 16? So let's do that real quick. I can't do this. 9 divided by 16 times, so that's 0.5625 times 200. Never use a decimal in your expected, okay? You can never use don't use the decimal number like the 0.5625, okay? You can use a decimal, that's fine, doesn't matter that much, okay? If you made this 113, it would be a slightly different number, but I still give you the credit for it, okay? 3 out of 16. Times 200. 200. This should all equal 200 as well, okay? So, now you do these patterns, okay? You have 4 values to find, and then you add them all together to get your actual chi-squared value, okay? But remember, it's 4 different calculations because you have 4 different values here, okay? So it would be observed 90 minus 12.5 squared over expected 55 minus 37.5 squared over 37.5, 43 minus 7.5 squared over 37.5. And the final one, I didn't squish it well enough, would be 12 minus 12.5 squared over 12.5. Okay. Do these 4 and you have your chi-squared value. Okay? I'm going to tell you right now, I just made up numbers here, but it's going to tell you that you found your expected number here. This is a very small number, that chi-squared difference, but these are all very different, and what you would say is actually something is happening here. Maybe it's a linked gene, maybe not, but it's you would reject the null hypothesis and say, no, something is happening here. This is not what I expected, okay? That you would have significant result, indicating that these are not in Mendelian ratio, what you expected. First part is this. I may give you a problem to solve first. What frequencies would you expect? Then I'll give you numbers and say, do these match this frequencies? And you'd have to do the calculation to find out. And I'll give you, obviously, a critical value to compare to. Angie, does that help? Again, I made this up off the top of my head, but that's the concept. Again, first you've got to figure out what is your actual expected percentages. Take this number, multiply your percentages, get those numbers, and then it builds itself. I will always give you the ability to find the chi-squared value to actually calculate that out. That's not a secret, okay? Don't mind if you look that up. Key here is that building it is the part, okay? All right. In terms of the calculation pieces, does anyone have more questions about this? Any of those bits? Make myself small again. Again, now you can go through and see all of these answers, compare yours. If you got it wrong, it'll tell you what the correct answer was, and it'll tell you what those correct information pieces are here, okay? What the actual thing I was expecting was. All right. Gonna minimize that. Okay. Again, for chapter 17, again, the ones we most recently went over the restriction mapping. The main thing with the restriction mapping is just you'll have a map already, and you're going to have to be able to identify which ones the restriction, let's say I'm going to give you like a picture or something, which ones restriction enzymes worked on, which ones, none of them, one of them, both of them, etc. Okay, complete digestion. And so comparing those, but basically just mark out, okay, if it cuts here, here, and here, then what would I see if only one of them cut? What would I see if both of them cut? And just make sure it all adds up to the end number. And then where would you see bars at? Base pair lengths. DNA fingerprinting and Sanger sequence, go back to the thing I show you how to do it. Go back to the videos. Also that video with the DNA fingerprinting and the activity really help with practice for that. Okay. So we talked about those pieces. Anyone have questions about the chapter 6 and before? It's been a little while since we worked through all of those pieces. We've referenced them, but anyone have more questions about combining, say, aneuploidies and inheritance or anything like that? Or want me to do one of these, like, what if it's an epistasis plus a codominant, like we talked about with the Bombay phenotype? Or the codominant and the multiple alleles, or the when we talked about the plus minus plus the ABO and how that makes things vary a little bit. If you have multiple alleles and you have the positive negative, you don't get this 93:3:1 because of that variation. Sure, Bombay phenotype. Back off for 1 second. I'm going to make myself big again and write it on the board. And recording this, you'll be able to watch me write it on the board several times if that's what you need. Okay? All right. Big again. And big. Okay. Next. We've got A, B, O. If you have an A, that's the A carbohydrate, B carbo, no carbo. Bombay phenotype has to do with a separate one. O, well, it does not have a carbohydrate. So for each one of these, so here's this square, here's this triangle. O has a place for the carbohydrate to attach, but no carbohydrate. Bombay, okay, has to do with the place for the carbohydrate to attach, okay? There's a separate, so if you have no place for the carbohydrate to attach, and you have an A allele, it'll come out like this. You have a triangle, nothing to connect it, no carbohydrate can attach to it. Even though you have the ability to make the carbohydrate, it cannot attach to your blood because you're missing the connector. That's the Bombay allele, the Bombay phenotype. If you have two carriers for that, then you will have the Bombay phenotype, which means your blood will look like O, but you cannot take O, A, or B. Okay? help a little bit. All right. So the complication with this is that someone with the Bombay phenotype, you can't always tell what their actual genotype is unless they have children, or unless they, you know that their parents were carriers. So you have an idea of their actual. So if you have an AB and then plus O, that's the parents. If you know what the parents are A, B, and O, and you have a child with the Bombay phenotype, AB plus O, can that ever make O? No, it cannot make O unless they have the Bombay phenotype, okay? So you can look at, for the carriers like this, what is their chance of having an O child? Well, it's only it's going to be actually a 25% chance actually they're going to have an O phenotype child, even though it's actually the Bombay type, okay? Because anywhere where you have these two H's come together. Okay, but that's the concept. And this will override a presence of an A or a presence of a B because it doesn't connect. All right. Other concepts? Okay, we're just about at the end of where we're supposed to be. We do have a planned Zoom activity as well. We're going to go through the current homework that you guys did. That was due yesterday. That was the extra credit homework. I know many of you did it anyways. So again, this is the bonus part. So we'll get on Zoom and go through that. But hopefully this helped people with a couple of the complex pieces. Again, if you have questions about any of the things that you see in the calculation bits, you say, you know, I remember I did this on here. Okay. Faye, if you can't play on the PC or the Mac, again, it requires a specific, I was hoping because it wasn't Flash, you'd still be able to play it. Okay. If you can't play it, what I'll do is I'll make a short video, okay? Should take me about 10 minutes to play it. So after we're done with our homework discussion, I'll make a short video and I'll post it as well. You'll have to watch the video, and I'm going to go through and do the parts. But what I won't do is tell you what the actual answer is at the end. You'll have to send that to me, okay? So I'll do that, and if you can't play it yourself, which is more fun to play yourself, if you can't play it yourself and make a short video, I'll get to the end, and you guys will have to tell me who the killer was, okay? But you'll have to watch the video and participate that way. All right, let me away here. Everybody gets a break. I'm going to. Computer's all confused now. Okay. Forgive me while I fuss for a second with everything. And I'm going to turn the. Go back here. Make myself small so I don't have to remember later. All right. Well, I will see you guys on the Zoom call. Remember, this is our last class. It's been great having everyone here. Again, feel free to open this back up and run through everything again, but we'll go through the homework. It's part of Zoom. I will see you guys in a few minutes. Let me put the Zoom post up. I just figured to do that before I go. I can never remember it. Let me just pull up Zoom. Okay. Here we go. Heska open this. All right, thanks. Let me pull up the actual invitation to the Zoom. I guess the same as before. Go here. I'll see you in the room in about 10 minutes. Okay. Turn off OBS. See you in a few minutes.